RNA helicase is involved in nearly every process in RNA, from structuring RNA to unwinding double-stranded RNA to providing innate immune responses. Unsurprisingly, RNA helicase can influence many diseases and RNA-based therapeutics. Today's podcast guest established one of the largest RNA helicase research groups in the world, gaining insights on just how a helicase relates to health and disease. On this episode of RNA Insights, we talk with Dr. Eckhard Jankowsky about RNA helicase's impact on health, transitions from academia to big pharma, and how the past 3 decades of RNA therapeutic research will influence the future of RNA medicine. Welcome to RNA Insights. Thanks again for meeting with me today. But just to get started, a little bit about what you do and how you got to where you are. Why did you initially decide to study chemistry and biochemistry?
Yes, it's always hard to put your finger on it. So there wasn't really like a kind of a pivotal point in my life where upon I decided to become a chemist. So it
Mm.
was more kind of in high school. So chemistry always seemed to make some sense to me. It was kind of cool to get a rational understanding of how things in the world would work. I grew up in East Germany, and science back in East Germany, like chemistry, it wasn't very politicized. So it was also something to escape kind of a relentless politicization of everything. So there was a practical consideration there. But chemistry in Germany is a big thing. So that seemed to be kind of a thing to me. And I should probably also mention that. So initially, it wasn't exactly chemistry that I wanted to actually study, but more food chemistry. I was very the kind of food aspects there. But that being East Germany, when I actually started to study, so it's a food chemistry, that track that was already on every other year and not the year where I actually started. So that was close enough with the chemistry. And then I essentially stuck with chemistry. And the biochemistry part, it was not really something that was focused on during the studies.
Mm.
Yeah, so, I mean, chemistry where I studied in Dresden was very, very traditional chemistry. So there was no bio. The university where I studied was a technical university, and they took that very serious as technical. There was absolutely no bio, which is really hard to believe if you look at Dresden these days. Dresden is a real big center of all kinds of bio stuff, and there was
Mhm.
absolutely nothing in the entire city back then. But they had an institute for wood and plant chemistry, and that's where I did my undergrad thesis. I studied essentially peroxidases, superoxidismatases in trees. So the idea was to figure out wood damage before it would actually be obvious. So that's how I got into the bio. And then during my PhD thesis, that was in so when I started doing that was in '93. And by that, the Biochemistry Institute had been founded. And I was one of the first grad students there to actually study there. So that's where I started to actually work on RNA. There was also very, very little guidance. I was definitely the first person who worked on RNA in Dresden, and at least from kind of a chemical point of view. And so I actually worked on hammerhead ribozymes. And the reason why I worked on hammerhead ribozymes was because you could make them chemically, and we had the synthesizers. They were small enough to synthesize them. That's how I got into RNA and biochemistry. Be a plan. It was, like a lot of coincidences, I guess.
Fair. It seems like that happens for a lot of scientists. You mentioned that you were the first person to go into RNA there. Did you have any sort of mentor at all, or were you really just kind of figuring it out as you go? You're like, "This is my research. I'm interested in this. I have the tools to use it. Let's tinker around and see what we can do."
So yes, that was the biggest part of it. But I did travel around Germany and asked a couple of other people who had done this before. RNA and especially chemically synthesized RNA. That was very exotic in '93. There were really only a couple of people, but they were all very nice. And Germany is not so big, so I got to know a lot of people, and that's how I got essentially started. But on site, it was essentially just me. So
Wow.
it was good. I mean, I kind of liked independence, and it
Yeah,
was a good fit.
cool. Obviously, many of your publications are about RNA helicase. Did you initially start looking at helicases, or did you get into that further in your RNA research career?
No, well, so the Hitty case is was as probably everything in everybody's science career was also kind of a coincidence. So right before I finished my PhD, I applied and I got a position at Anna Marie Pile's lab at Columbia as a postdoc. That lab was a ribozyme lab. Like, made sense since I had worked on ribozymes before. But Anna asked me of whether I wanted to look at these RNA helicases or RNA protein interactions. So that it was not something that I had done before, and it seemed interesting to me. So RNA helicases, there are all over RNA biology. So wherever you have RNA, you usually find some RNA helicases as well. The largest group of enzymes in RNA metabolism that was known back then, and there was a lot of push. It was pretty clear that they would do something that's relatively pivotal in RNA biology, but it wasn't really clear what this was and how they would do it, and there was a lot of interest there. It wasn't really understood how they would work at all, and I thought, "Oh, maybe we should give it a try and see whether we can find something out." And she asked me to do that, and that's kind of what I did. So that's how I got into RNA helicases. Um, in Anna's lab, we worked on so first I started at a lysosomal helicase and at a viral helicase together with Christian Gross from Stuart Schumann's lab. That was a great collaboration. They were also local. Columbia is on the west side of New York City, and Stuart and Christian, they were on the east side at Slow Kettering, so it was just cross town. So it was a fantastic collaboration. It was a fantastic system to get people at.
Dr. Anna Marie Pyle founded the lab where Jankowsky worked with her in 1992 in the Department of Biochemistry and Molecular Biophysics at Columbia University Medical Center. The first lysosomal helicase that Pyle and Jankowsky studied was the DEAH box protein PRP22. I linked to their first paper on the helicase in the show notes. It's called The DEAH box protein PRP22 is an ATPase that mediates ATP-dependent mRNA release from the spliceosome and unwinds RNA duplexes. To make sure that all listeners are up to speed, do you mind just kind of broadly talking about what RNA helicase is and what it does for RNA itself in the human body?
Yeah, so RNA helicases are a large class of enzymes. Actually, it's the largest class of enzymes in RNA metabolism. In our eukaryotes are like about 60 to 80 members. And so what they do is they RNA unwinding function, but they don't all unwind RNA, and certainly not all in kind of a classical helicase sense. They're essentially ATP-dependent RNA remodelers or binders. So that's a more comprehensive, all-encompassing classification. So they bind and remodel RNA or RNA protein complexes in an ATP-dependent fashion. Then when I started working on this, that wasn't really clear, but they're really more kind of ATP-dependent RNA switches, more like G proteins to some extent, even though some do clearly unwind. Involved in essentially all aspects of RNA metabolism, from transcription, export, translation, RNA. So I'm not aware of actually any large process in RNA metabolism that doesn't involve one or sometimes a lot of helicases. And they are very, very conserved. They have a very conserved core. So structurally, they are super, super similar. There are, differences with respect to certain sequence motifs, and they classify into certain families and subfamilies. And our work contributed quite a bit to that understanding. So there's some functional diversity there, but they're essentially the ATP-dependent drivers of RNA metabolism. I think that's a reasonable description.
The primary family that you worked with was the dead box family, correct? Or are there other families that you worked with the most?
I did work with other families. So dead bugs, helicases, so they are, so it's a very memorable name that was actually coined by a Swiss scientist, Patrick Linder. And where that comes from is actually a conserved amino acid motif that reads DEAD in single letter code. So it's a good name. If you hear once, you definitely remember. Many are actually essential for life. Want to make sure that the dead box helicases do function, because otherwise you literally end up in the eponymous box. This is actually the largest family of helicases. So there was more choice to actually study these, because when I was setting up my own research group, The idea back then was sort of a helicase, an RNA helicase, and the helicase that we had studied in Anna Palt's lab, that was certainly consistent with the classical helicase motor thing. The first paper that I published there was essentially stating the fact that this was a molecular motor, and it clearly was. I mean, we looked at like how it translocated and quantified this and all these kind of things. It was a classical helicase.
Together with Dr. Christian Gross from Dr. Stuart Schumann's lab, the viral helicase that the trio researched was the DEXH helicase NPH2 from the vaccinia virus. I linked to their first paper together. The DEXH protein NPH2 is a processive and directional motor for unwinding RNA in the show notes. I also listed Jankowski's full list of publications.
Usually because they have duplex and then they have a single-stranded overhang. And that, that single-stranded overhang has to be in a certain polarity. So many helicases require that on a 3' end. If you put on the 5' end, that doesn't really unwind duplex. That's what the classic helicase does. But for dead box helicases, they didn't really care. I mean, you had to have an overhang, but it didn't really matter where it was. That was unusual. And I thought, oh, okay, well, let's see what is going on there. And that's essentially what the motivation was to look at dead box helicases. Then later on, we discovered all kinds of non-intuitive things that these enzymes do. So they do not behave like like translocating helicases.
Yeah. What are some examples of that? What are some of the notable things that make them behave differently than traditional helicases?
So what dead box helicases do, and I'm not aware of really any exception, is they do separate duplexes, but they do this in a very kind of localized fashion. So essentially, what they do is many oligomerize or they bind to cofactors, and then they directly load on the duplex. They essentially tweak the duplex, and that separates them. That's all there is. So they're very good at separating short duplexes and really can't do very long ones unless they oligomerize along there. That's precisely what they designed to do. They kind of large RNA assemblies. You don't necessarily want to unravel all these things, right? And you do rearrangements in RNA metabolism. So you want to make local changes made. And that's exactly what the mechanism enables these enzymes to do. So yes, so we termed this unwinding by local strand separation. So going on the duplex, the tweaking this a little bit and in an A-dependent fashion, that's essentially the mechanism. So it sounds trivial, and that's how it always should be. Right after you've discovered something, it should be trivial. So there's no miracle.
Yes.
It was a process to come from the notion that you always had to have this movement to enzymes that essentially don't move at all, affect these conformational changes.
To reiterate, most helicases unwind RNA duplexes by moving along the length of the RNA strand in a specific direction, usually from the 5' to 3' end, but some move 3' to 5', and unwind RNA as they go. Dead box helicases, though, unwind RNA duplexes locally. Rather than moving along the RNA, they unwind RNA duplexes by binding to the small region that needs to be unwound and then release the RNA after unwinding. So obviously RNA-based therapeutics are becoming more and more of a thing. That's really the basis of this podcast is looking at how different RNA research and science and biology relates to therapeutics and making them more effective. How do RNA helicases, especially dead box, impact therapeutics? What are some of the main things in biology that can make therapeutics better?
Helicases themselves, they probably interact with some of the RNA therapeutics that you put into the cells. At least with mRNAs, I'm fairly certain you need a couple of helicases to do their jobs there.
For sure.
So far as iRNAs, I'm not so sure whether there's really a helicase involved once you put endogenous RNA into the cell. But for mRNAs, I'm sure they're involved, but not in an explicit way. There's not really a kind of a direct connection to RNA therapeutics. I'm sure we get to how that connect. Well, there's really connections, but there are quite a number of implications with respect to kind of medical implications for RNA, I guess, because of their ubiquitous role in RNA metabolism.
Did you explore any of those throughout your research, or had you been more on the biology side primarily, or biology of the RNA as opposed to medicine?
Yes, we were very, very focused on fundamental mechanisms. But we were always open if somebody had kind of a problem with a medical problem with a helicase. So we have a couple of papers. We had a collaboration with group who worked on MDS, so myelodysplastic syndrome. That's pre-leukemia. It's often found in the elderly. And one of the drivers for that is the helicase. It's a spliceosomal helicase. So we did quite a bit of work on that one. And then there are a couple of other helicases. So we worked on one helicase quite extensively called DDX3. That has been implicated in a lot of different cancers. It plays a role in translation initiation. So you always need protein synthesis and in a lot of cases. And we had worked for a very long time on the yeast orthomorphs. So they're very, very similar. And they seem to do quite similar functions. They also fairly non-intuitive ones. And so there was a lot of connections. DDX3 is also involved in developmental issues. There's actually DDX3X syndrome. There's a very active society. So, I mean, there are not that many people who are affected by it. It's mostly so DDX3X is on the X chromosome. It seems to be very, very important, but it's one of the proteins that actually escapes the X chromosome silencing, since females have like one X chromosome more than males. I've got to do something. And much of the X chromosome is usually silenced, but not all of it. About 10% escapes the silencing. And DDX3X is one of these. The developmental processes that are significant implications, right? I mean, it's early stage developmental things. So you can't really do anything about it. Once that is, gone not right, your development is derailed. And it affects mostly females, surprisingly enough. You think they should have two of these things, but there's reasons for that. This one protein that we studied, there's all kinds of further implications. There's now probably thousands of papers on that protein.
As Jankowsky said, DDX3X is a gene on the X chromosome that encodes a dead box helicase. When this gene is mutated, it disrupts this helicase's ability to function and properly unwind RNA, which can impact brain development, called the DDX3X syndrome. This syndrome occurs 95% of the time in females on only one of their X chromosomes. Scientists believe the syndrome is lethal in males in utero since it affects their only X chromosome. The mutated gene's impact on protein expression is also associated with some cancers and viral diseases, so addressing this mutation is of therapeutic interest. Some of these proteins that are being identified for causing developmental issues are different of disorders and diseases. Are we at the point in research where we're like they are identified, but we're still figuring out next steps, or are researchers finding different medicines and ways to address it?
Yeah, that's how it often is for therapeutic. If you try to therapeutically target it, always depends on the specifics. With respect to these neurodevelopmental processes, you have to obviously deal with this before it really manifests itself. Some of the early antisense drugs did exactly that. They were total game changers for therapeutic implications. For these mutated proteins, I don't think there's really anything at this point in time that people can really do. For this particular protein, DDX3, there are a lot of things coming together. That protein also is involved in kind of cytoplasmic granule formation. So what that essentially is probably some kind of stress response where a lot of proteins are then put together in extracellular granules, stress granules, or other granules. If there's something that isn't really working correctly, then this granule formation is accelerated. Essentially, you have a stress response when there's no stress. That's an issue. That seems to be a big part of the disease development. For some of these indicators, it makes it counterintuitive to actually interact with them. Normally, you would say, okay, if there's something wrong, I want to inhibit their function, right? But that would make things sometimes worse. So you can't really deal with this in this kind of traditional manner. There's ways potentially to attack this. Before I actually crossed over into industry, we started working on this a little bit. There's one paper about that. We have to see that you attack these kind of interaction domains, but not the catalytic domains.
I've linked to this paper. The viral protein K7 inhibits biochemical activities and condensate formation by the dead box helicase DDX3X in the show notes. As the title suggests, the paper found that the protein K7 in the vaccinia virus limits how DDX3X associates with stress and aberrant granules by binding to the helicases N terminus. This can help reduce DDX3X syndrome.
And you can do that. There's ways. Actually, viruses have figured out how to do this. They can take a page from what they have already tried.
You mentioned both industry and virus here, which seems like a great transition to talking about Moderna. Um, for you personally, obviously you spent over 2 decades in RNA research on the academia side and then made the switch over to being VP of RNA science at Moderna. What inspired that change? What led you, especially I'm assuming the pandemic, I know you kind of were at the tail end of that, but I'm guessing it did relate to it somewhat. What
Yeah.
inspired that change?
Specifically for me, in early 22, February, early March, I got a call and they asked me whether I would consider leading the RNA science department. So, so that was very surprising. We hadn't really like worked in any explicit manner on RNA therapeutics, but it made a lot of sense over the years. We had worked on a lot of like things that were very, very relevant. I mean, in 22, it was very hard to say no, right? That was literally coming right off the top of everything. I thought, well, that's definitely one of the coolest jobs in RNA that you can. So I said yes. And then in summer, I switched to Moderna from academia.
Cool.
So I went down the lab and did the cold plunge into industry.
How was that transition? You mentioned a cold plunge. Was it a pretty fun but jarring transition, or was it relatively smooth?
No, it was great. I really loved it. Things did change over the years that I was there. I don't know exactly how the Bell Labs were, but I saw there was kind of something like the Bell Labs for mRNA therapeutic development. There was a very expansionary phase there. The idea was really to take very ambitious look at how to expand the scope of mRNA therapeutics. And I saw that it made all a lot of sense to me. Business conduct is very, very different from academia. But with respect to it was science, it was very, very, very rigorous science because if you do something, it has to work. Not that you wouldn't do a rigorous science in academia. So the paths are very different. I thought that was first of all cool. And then of course you could move a lot faster than you ever could in academia. Once something was set for the next quarter or half year, then you would do it. There were this professional scientists. They got a lot of stuff done. It was fantastic. The surrounding teams were incredible. It was very, 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 very good. So,
Also,
so then things changed a little bit, because of all the headwinds that I'm sure everybody's aware of. But I have to say, so the first, almost like 2 years that I was there were incredible.
you mentioned expanding mRNA therapeutics. Are you able to elaborate on that at all?
So essentially, so what we did at Moderna was expanding mRNA therapeutics. There are some limitations with respect to how much protein you get off a given RNA. So there's
Mhm.
always a push to get more of that. So that was a big goal. And then you want to make this RNAs a bit more specific, so that they only work in certain contexts. So that was something that we focused on. Then there were a lot of things that needed to be tended to from the production process. That was also something that we focused on quite a bit. And there was analytics that we developed to optimize things better. Essentially, optimization for our mRNAs, that was the overarching goal there.
Was going from being in academia to industry, I'm guessing manufacturing and optimizing, especially for the manufacturing side, becomes a larger thing. Was that interesting to you? Was that different compared to being in
Um,
academia?
Well, it's definitely different but it's a scientific problem to solve. It's not one that's presented by biology. And as a matter of fact, to some extent, it might be tougher than if you choose something. Eventually, in academia, if something becomes too hard, you can always move something to something else. But that obviously doesn't really work if, if there's something that has to be sorted out for a production process. So I have to say, I found that equally cool and challenging. And it's obviously very important to want to contribute to a product, and that product has to be good. And so I didn't see a problem with that at all. Essentially, whatever comes your way while you're doing this, you do it. And there are many ways to apply the RNA knowledge. To some extent, it's probably conceptually easier to solve because we usually was dealing with some not like open systems as you do in biology. Right? In biology, you never know. There's some things that comes from left field in a totally unexpected manner. They haven't paid any attention. That usually doesn't really happen if you deal with something technical. Sometimes these are tricky problems to deal with, and they require a lot of dedication and focus. But maybe it's different from biology. It's not as open ended.
Yeah, that's very interesting. That makes sense. Pivoting then from Moderna, now you are working in like consulting and strategic advising.
So the
How has that been?
consulting, the consulting wasn't exactly voluntary, as I'm sure you know. So there was a lot of headwinds. So at the end, they had to adjust, and the adjustment was accompanied by a very large reorganization that happened like last summer, last fall. And unfortunately, it fell heavily on research or development. So my position got eliminated. So I got to do something. At this point in time, it's tricky to find something more long term, but I'm still looking. That's something that I am very interested in. In the meantime, consulting, what you can do, that's what I'm doing. It's quite interesting, I meet a lot of people.
There. It seems like a very creative way to use all of the skill set you have in the meantime. That's a cool way for you to use all of the skill sets and experience you have. But speaking of the experiences you have, taking for these last few questions here, kind of taking a step back and looking at your career overall, what is one or two or three of like the biggest discoveries or breakthroughs in RNA that you've seen in your career that you think have made a big difference or are going to make a big difference in the next 4 or 5 years?
So I've seen quite a bit of. So when I started, there were no microRNAs. That's very hard to imagine. But actually, when I started, the first PCRs that I did, there were no pleated lids. You had to now you put oil or a few things. That was the way to do things. So the things have come a long way. But if you have to pick one thing that is really the most game changing thing, I would probably say it was probably sequencing. That has opened up a totally new way to look at RNA. I think it allows to look at RNA biology with a perspective that is useful, right? You can study one gene and then there's things you learn, and often it's transferable, but often I'm not sure how long it would take to get comprehensive pictures. With sequencing, that really changed the perspective. We did a lot of sequencing work in my academic group as soon as it became available for normal mortals. And so there was around like 2010 or 12. But what we used sequencing first was actually doing the in vitro massively parallel kind of screen, essentially looking at not one substrate for actually it was for protein binding. Our first groups there, not just looking at one substrate, but at all of them. There was like thousands, so that was cool. And then you got a whole different perspective of how proteins discriminate between different substrates. So that has really fundamentally changed.
I've linked to one of Jankowski's initial papers using sequencing to screen substrates, called Hidden Specificity in an Apparently Nonspecific RNA Binding Protein, in the show notes.
With that in mind, I would probably put the finger on sequencing and whatever grew from there, like lip, riboc. I mean, it's very hard to imagine now with how that has shaped on RNA biology, how that actually was before. That I think was probably the biggest game changer, and everything that data analysis, model building, and all these kind of things.
It's a big umbrella that really does help start closing some of these gaps and just make everything quicker to do, more accessible. What then is one thing that you predict is going to happen in the next like 5 to 10 years, the next technology or modality that will take over, help make the next breakthrough in RNA medicine?
Yeah. So for RNA medicine, yeah, that's always a good question. That is not so, was obviously not straightforward to answer, because if I knew, I would probably kind of, already be working on it. Let me kind of focus a bit more on the mRNA side there. So even though that has gotten a lot of attention, and obviously more than we would like, it's not always the most constructive one. But that is really, it's essentially just the beginning, I would say. So there's still a lot of room for improvement. So if you look at what currently is being done with mRNA, there's a lot of other options that can be pursued. Nothing is trivial in that field. So this is a complex therapeutics, but you increase your understanding and you increase the way you can deal with this. Something where I would predict quite a bit of progress, despite the unfavorable
Yeah.
kind of view on these things. But, that's a multifaceted cultural issue. But scientifically, that is, I think, something that there's a lot of potential. And there are a lot of other RNA related modalities, like
Mm.
aside from siRNAs. I mean, there are like editing modulators. It's a bit more specific for what these modalities can be used, but it's a very cool use of RNA biology for a specific therapeutic outcome. So that's, I think, is exciting too.
For sure. It'll be exciting to see what happens. Last two questions here. What is one of the most challenging aspects of your career in RNA research and industry, et cetera?
Well, at the moment, I mean, yeah, I think it would be nice to have kind of a more permanent
Yeah.
base. So that would be obviously nice. But in general, it might not necessarily be true for RNA. So I think the RNA community is actually a great community. I really enjoyed being there. Very supportive. It was never so big.
Mm.
So you got to know a lot of people, and a lot of people have actually stuck with it. So it's not like, that people floated in and out, and then, after 10 years, you wouldn't really recognize the scene anymore. So that really has not happened. So there's people who usually got into the RNA community usually stayed. And to some extent, that's semi-true from what I gather in the mRNA therapeutics community, at least that's the early development part. There's a meeting that is jointly organized by CureVac, BioNTech, and Moderna, the mRNA health
Mhm.
meeting. So this is very scientifically focused, and people like it quite a bit. Just to illustrate that there's a strong scientific community.
The annual meeting is the International mRNA Health Conference, a nonprofit event co-organized by Moderna, CureVac, and BioNTech that brings together industry leaders and academics in the mRNA medicine space. In the show notes, I've linked the information page to the 14th annual conference, which will be held in Boston from November 17th through 19th, 2026.
But with respect to challenge, that's not specific to RNA. So if you do science, you always confront people with something new and a lot of rejection. So
For sure.
I mean, I guess there's a lot of rejection anyway when you interact with other people and want something from them. But that is something that you got to get used to. And you have to, otherwise, probably not going to make it very far. But I think that's an eternal challenge, right? I mean, when you develop an idea and it's a lot of work and then you present it. But, you know, you have to deal with it.
For sure. Last question here. What's your favorite part about working in RNA research and industry?
So I always thought it was somewhat exotic. At least when I started, it was super exotic, and it, it maintained. I think that kind of, that kind of edge. I thought it was always not a particularly large field, as I mentioned already, but it was often contributing things to biology or to medicine now that you're really going to get from anywhere else. I thought that they're kind of a little bit off the beaten path thing that always I found very, very appealing. And as I mentioned too, I mean, the community always matters. If it's welcoming and people are interested in what you're doing and you're interested in what other people are doing, that's a good basis for working in the field for a long time.
Thanks again to Dr. Eckhard Jankowsky for joining this episode of RNA Insights. Before we leave, here are a few recent pieces of RNA therapeutics industry news. Arena presented preclinical data on AIR-001, an RNA editing therapeutic candidate for the lung and liver affecting alpha-1 antitrypsin deficiency. The first in vivo results suggest that a single dose of AIR-001 in mice reached 59% editing to help correct alpha-1 antitrypsin levels, and longer-term dosing further treated lung and liver injury. Orna announced its first in-human study of and presented preclinical data on TI-0032, a circRNA-based in vivo CAR T therapy to treat recurrent and refractory autoimmune diseases. The early data suggested TI-0032 successfully reduced autoimmune disease effects in human immune cells, humanized mouse models, and non-human primates. Aviado Bio announced preclinical data for AVB406, a single-dose gene therapy utilizing the company's VMix RNAi platform to treat Alzheimer's disease by silencing the MAPT gene. The data suggests the therapy was successful in crossing the blood-brain barrier, achieved gene knockdown, and was tolerated by cells. Thanks again for listening to this episode of RNA Insights. Tune in next month to hear us talk to another RNA expert.